Good morning and welcome to the Friday morning seminar. I'm supposed to talk about our contribution to the OpenCall2 consortium, and the talk is entitled Metabolic Responses to Phosphate Deficiency. As I said, this is a project part of the OpenCall2 project led by Gabriel Scharf, crop responses under phosphate deficiency, integration of plant physiology and sensing and modeling. And the obvious compound we are talking about is phosphate deficiency. Other partners of this consortium are Pang Yu, Andrea Schnepp, Matthias Vesuva, and further collaborators. Can you actually see my cursor here? Yes, okay, thank you. So we have a core consortium of 5 members and plus additional co-workers. And the focus of the work is to study, in general, the phosphate starvation response in 2 crop plants, barley here depicted on the left, and corn, maize on the right. Okay, why would we bother about phosphate? I assume that many of these slides or similar slides you've seen in Gabriel's talk, because he is a major contributor to the phosphate starvation research area. So phosphate is an important macronutrient. It is highly abundant in plants, 5 to 20 millimolar. That's quite high. So it is an abundant compound of plant. It's important. We find orthophosphate like this anion here, which is inorganic, PR, abbreviated PI, mostly stored in the vacuole of the plant. But in addition, we have organophosphorus compounds, which means this part here bound to organic molecules. And if you look into a leaf, you can find that about 1/3 of the phosphate of the organophosphate, of the organically bound phosphate, is bound to DNA, RNAs, nucleic acids. So genetic material in a wider sense. 1/3 is bound to membranes, phospholipids. And the last third is bound in nucleotides and sugar phosphates, so in metabolically active compounds where phosphate also plays a big role. These are the major pools of organic phosphate, plus the inorganic phosphate in the vacuole. Phosphate is limited in the soil because if it is there, it might be insoluble or bound to soil particles. You can find it and harvest it from rocks, phosphorus rocks, which is a limited resource. And if you do so and apply it to the fields, you need to be careful not to do too much, because you easily run into water body eutrophication, because eventually the phosphate is washed out into the lakes and rivers. And then if phosphate is taken up to the plant, it's either done directly by the roots, phosphate taken up from the roots, moved to the vessel, through the plant in the form of inorganic phosphate, PI, orthophosphate, or organic phosphorus compounds, as I just described, and then find their way to the leaves, to the shoot parts where they are incorporated into lipids, nucleic acids, and so on. There's also an independent, indirect way when plants undergo symbiosis with fungi, which helps them to mobilize phosphate from more distant parts of the soil, more deeper soil areas, for example, because this mycorrhiza extend to further parts and live in symbiosis with the plant and provide phosphate to the plant. Okay, we'll also talk a little bit about phosphate sensing. This is work where Gabriel also contributed a lot. So we obviously put the plants under phosphate limitation to trigger the phosphate starvation response, PSR. Here in the center, PI starvation responses. And the sensing in the plant, when they grow under low phosphate, the plant senses the low phosphate. The transcription factor PHR1 is a central one, which induces the phosphate starvation response gene expression. So dozens of genes are change their expression, which results in the phosphate starvation response. affecting growth. I mean, as expected, you will find reduced root growth, seed yield, proliferation of roots. Photosynthesis is impaired. This is not our, and obviously the avascular mycorrhiza fungus symbiosis is established, but this is not our primary focus. What we mostly look at is metabolic changes, for example, prominent changes in anthocyanin accumulation. You also look at secondary metabolites in the roots and the lipid changes, which are important to provide phosphate to the plant. Okay, this sensing part has been elucidated in Gabriel's group recently, and we also have a side project on that. Because it is not directly the orthophosphate which is sensed, but it's this more complex inositol molecule with several phosphates bound to it, 8 in this case. This is produced under high phosphate by VHA1 and 2 enzymes which produce this compound, which is then the sensed molecule, sensed by these 2 proteins, PHR1, which I already mentioned, and FBX1, a helper protein which binds to PHR1. And under high phosphate, the phosphate starvation response is suppressed. You look more into detail. So under high phosphate, this inositol containing 8 phosphates is produced, binds to this complex of the 2 proteins. This protein remains in the and cannot interact with DNA. And this reaction is catalyzed by VH1 and 2. But under low phosphate, the reaction is reversed. 1 phosphate is cleaved away, and this complex falls apart because the inositol with the 8 phosphate is gone. PHR1, the transcription factor, is released because complex falls apart and binds to the DNA and triggers the phosphate starvation response gene expression. Okay, the outline of our study. We first start with the metabolic to establish metabolic responses in barley. Our first model system is barley. We also look into the shoot and into the root exudation, but this we do with mice then because it's in collaboration with Panyu. We generate mutants of these 2 enzymes, VH1 and VH2, together with Gabriel. We look at changes in gene expression in order to identify phosphate-dependent promoters, which we need to produce smart plants being reporter plants, which express a gene which gives rise to a color, ruby, and this idea goes back to a collaboration with Amin Jameh. Okay, and the model systems are still barley and rice. We start with barley and look, this is experiments that Magdalena Kuskowska started in the lab. Phosphate starvation of barley. Barley has, Magda very fast figured out that it is not possible to just take the entire leaf for these experiments because the different leaf parts respond quite differently. Therefore, she separated the leaf into 3 zones. The leaf tip, which is the oldest part, center, and the youngest part, where the leaf grows. This is the elongation zone. And as you will see in a minute, the metabolic responses differ in these 3 parts. We grow the plants in hydroponic medium with or without phosphate. We then do a time course after initiation of phosphate starvation and then measure different parameters. Yeah, this is looking at phosphate plus phosphate and then the phosphate starvation response. Plants taken to phosphate starvation in the shoots and roots. And in this case, in the leaves, we measured leaf tip, middle part, the bottom part, which is the growing part, here elongation zone, leaf tip. So first phosphate in the leaves to see when the phosphate starvation starts in the leaf, and you can see that after 9 days, phosphate is suppressed, phosphate content is suppressed. So after 9 or 12 days, we have a significant phosphate starvation response. In roots, even earlier, already after 6 days. Okay, and then measured compounds which are known to be affected by phosphate deficiency after 9 days. Chlorophyll is down in the leaf tip, so in the oldest part. And similarly, anthocyanin accumulates also in the leaf tip. The other parts of the leaf are not so responsive. Okay. Then looking at lipids, we expect that phospholipids, which contain phosphate, are degraded under phosphate deprivation, mobilizing the phosphate for nucleic acid synthesis to maintain growth. And these lipids should be replaced by glycolipids, sugar-containing lipids. For example, galactolipids like this one here, DGDG, and sulfolipids with a sulfonic acid head group, which is very special. Here we have the glycolipids, the sugar lipids, and here we have the phospholipids. Glycolipids, and we always compare 9 days plus minus phosphate, 12 days plus minus phosphate. MGDG doesn't change, so this is known, but if you compare green and brown bar after 12 days, this galactolipid goes up and the sulfolipid also goes up. This is good. And at the same time, the phospholipids, all of them actually, all 4 that you find in barley leaves are decreased. So indeed, we have a shift from phospholipids to these 2 lipids in the phosphate deprivation. I find this quite interesting, the sulfolipid result. Sulfolipid is one of the few compounds in the leaf that contains the sulfonic acid head group, and it has been shown that this is active, has active vibrations in the near-infrared area. Here CS and CSO bounds show specific bands in the near-infrared spectrum. And this might be an idea for the future, the outlook, to look into the possibility whether this molecule can be taken as a reporter and might give rise to bands that might be visible in the near-infrared reflectance spectra, which have been recorded also with here, this taken from British literature. In strawberry, you can see spectra taken from leaves. The question would be whether this such bands could be observed here in the spectra by taking wild-type and mutants through such a study. That would be quite interesting, I think. Okay, another experiment that we have not yet started, but that we will do soon, is non-targeted metabolomics. I want to introduce this concept. This is looking for changes of unknowns, which will be important to characterize the phosphate starvation response. We take 5 plants, leaves, roots, whatever, plus phosphate, minus phosphate, do a lipid metabolite isolation, generate LCMS chromatograms, 5 chromatograms with phosphate, 5 without. Then we need to compare these 5 to 5 chromatograms. First, make an average of the control and the phosphate starvation. There's a software package available at the Scripps Institute. You just submit your data. This is done online. And the program then gives you a list of compounds which are different between plus and minus phosphate, unknowns. Here an example where we have high amount of this compound in the control and low amount in the experiment. These compounds can then be identified based on their mass spectra and can be used to describe and identify novel compounds which are important in the phosphate starvation response. So we will do this with different parts of the plant and compare leaves, roots, and also root exudates could be taken. This is for the future. And then I come to the root exudate program that is done with Pang Yu, because in his group, and this is now a mice experiment, it has been shown that certain secondary metabolites are exuded under phosphate deprivation, compounds like apigenin and luteolin, which are flavonoids, and we are still in the way to establish this method with the mass spec. Now, these compounds are exuded into the soil to attract bacteria to the root, which help and assist in phosphate mobilization to the root. So this is a very specific project. Okay, I then come to the VH1, VH2 story. We have established barley transformation in the lab, which is not so trivial, and using this method to knock out genes, targeted knockout by the CRISPR-Cas system that you have probably heard of in the media, where you can, at specific position in the genome, introduce mutations. And we do this for this very important pathway, the VH1, VH2 enzymes. We have two such enzymes in Arabidopsis, and Gabriel worked on that. And these mutants are known, and we have created mutants of the same two genes of the orthologous genes in barley. They're also two genes. The idea being to knock out one or the other, or in some plants even both, to create a double mutant, where this pathway has been blocked, where this complex cannot be built anymore, and the suppression of the phosphate starvation response is blocked. We expect to eventually identify plants which might hyperaccumulate phosphate. And Magda, who did this experiment, already created about 40 transgenic plants, which are growing in the greenhouse. And she has identified single VH1, VH2, and also double plants, all of them heterozygous in the first generation. So we need to go through several generations to obtain homozygous plants and homozygous double mutant plants, provided that they are viable. Once we have them, we can try to use them to train sensors and algorithms for PI starvation, and of course to characterize these plants on a biochemical level. Okay, this is one project. Then the next one deals with for the generation of the ruby plants. We need promoters which are dependent on phosphate. And as I showed that PHR1, the transcription factor PHR1, binds to promoters and triggers expression of genes under phosphate deprivation. In Gabriel's lab, Yisham did transcriptomic experiments, large-scale screening for phosphate deficiency inducible genes, and pulled off dozens of them. Magda did detailed analysis by qPCR on a subset, on a few of these genes. Here in orange, plus phosphate, in yellow, minus phosphate. These are qPCR expression experiments. As you can see, some of them she could reproduce, some not, for whatever reason. I mean, we have a slightly different system. We have hydroponics and so on. Anyway, she picked two of those, PAP3 and DSP1, which are induced under phosphate deprivation. And this is not shown here, which are not so much induced under any other conditions, because what we need are promoters of genes which are specifically induced under phosphate deprivation and don't change so much under all other conditions. And this we could figure out by looking into the other transcriptomic experiments that Champel did. Okay, so these are two candidates for the Ruby story. Yeah, Ruby, what is that? This is an idea that came up in discussion with Gabriel and Armin Jammet to produce crop plants, in this case barley, which can be taken as a reporter for phosphate deprivation. We have here a gene fusion, 3 genes. I mean, you don't need to know these abbreviations, and this is the pathway that these enzymes catalyze. They take tyrosine, which is an amino acid which is abundant and present in all plants, and through 3 reactions catalyze a compound called betalain, which is red. It is pinkish reddish, visible with the naked eye, and just accumulates at the site where it is produced. This construct is available. You can put it into barley. And we did that with first, in order as a proof of concept, with a constitutive promoter which is expressed everywhere and at any given time, U6, the ubiquitin promoter. And if you do this and create transgenic plants, that's what Magda did. You get, this is a transgenic barley plant, which should, the wild type would be green, but this plant is very reddish here at the elongation zone at least, because it expresses this construct under constitutive, with the constitutive promoter and is red all the time, which means that this ruby construct is active in barley. And the next step was then to use different promoters. That's what she did. So we have already PAP3 and TSP1 promoter fused to this construct and introduced into barley plants, and this is underway right now. During these experiments, we came to the point that we were not convinced, and we don't know yet, that the expression of these promoters would be strong enough to make a clear-cut difference. You can see also low, if you go to various phosphate concentrations, you might run into problems that the accumulation of the ruby might not be sufficiently visible. Therefore, Magda actually pulled this out, a publication from the Herrera-Estrella group, where he described a synthetic promoter of this binding domain as a hexamer, 3 tandems basically, fused to a minimal promoter. And this he did in Arabidopsis, and could show that when you have this hexamer, you can get an up to 48 times expression of induction of gene expression when you have a reporter fused to it. This was gas. So we took the same thing and fused it to ruby, and this is also the third construct underway right now. Okay, this is what I wanted to show. I'm now, so what the current state of the project, because we're still in the middle of the project, obviously. We did the initial establishment of the system, PI starvation response in barley. We can show that we see decrease in orthophosphate, induction of phosphate starvation response genes. If the typical accumulation of anthocyanin, chlorophyll degradation, replacement of phospholipids with non-phosphorus lipids, with glycolipids. We've established the barley transformation protocol and used it to generate the first ruby plants. The first one was the constitutive promoter, the others are underway. We are on the way to isolate the VIH1, VIH2 plants. This is underway. Outlook. Obviously we need to, and this will be quite a lot of work, to identify the VIH1, VIH2 plants. We need replica plants, independent transforms, and so on and so on. Obviously then do phosphate deprivation experiments and characterize gene expression and metabolic changes. Establish the smart plants, the different promoters. Measure the consequences of phosphate deficiency in shoot roots, different parts of the plant, root exudates, using targeted and in particular non-targeted methods. And maybe some of these molecules like sulfolipid might be useful to try them as a reporter for PI starvation using non-invasive methods. I would like to thank Magda, who did basically most or even all of the work. The Ruby construct was generated with help by Armin. Then I had lots of help and discussion with Gabriel on VIH1, VIH2. Nutrient deficiency experiments with Yeshambel. Here, the mice root exudate experiments are done with Pang Yu. I would like to thank my own group for the body transformation, Christiane Buchert and Marius Becker. And here, lipid epigenomic experiments are done with Mars Beck. And funding is from Open Call 2, obviously. Thank you very much.